All right, Project 6, the article in Project 6 with this very long title, but in short, you could say that it is about link rate generating long range sequencing information. So, practically, it's a technique to do haplotyping through generating linked read libraries. And the method in this article is called TELSIC, and it's from a startup company, Universal Sequencing. And it's a nice technique. It has only 4 steps before doing amplification and library preparation and then sequencing. And all these steps are done in one reaction tube. Very simple, very easy to do, and the whole process, I think it's you are done in 3-4 hours. Of course, 3-4 hours before you do the amplification and then library preparation and so on. And yes, and it's very easy to do this or perform this. It's practically mixing or mixing different reagents, waiting, and then mixing another reagent, and then waiting, and then the last step is to denaturing or to denature the enzyme that you have used in this system. So let's walk through all these 4 steps and see how this method works. So the first step is tagmentation. So we are using TN5 together with long genomic DNA, high molecular weight genomic DNA. Mixing this together with bar-coded beads, about maybe 10 million of these beads are added in this system. And each bead, each of these 10 million has a unique barcode on the surface. If you remember TN5, TN5 tagments the DNA, and while it does it, it incorporates adapters at the ends of the tagmented DNA molecule. And in this method, we are also using this TN5 enzyme to cut long genomic DNA, or as it is called, high molecular weight genomic DNA. In the Illuminas protocol, we are using a concentration of TN5 that generates fragments that are around 300 bases. And this DNA that we are using is not high molecular weight, it's relatively short. But in this method, you are using very long genomic DNA. Let's say it's around 10 to 100 or more than 100 kb, which means that we will have a lot of, and these are TN5, a lot of enzymes that will bind to this long genomic DNA and cuts it. And the distance here between these is around, let's say, 600 base pairs. So we have a very long genomic DNA, and there are a lot of TN5 molecules that will tagment this. One thing that I'm not sure if I have mentioned this, but TN5 binds very strongly to genomic DNA. So when we cut this, it's not really fragmentation. The TN5 cuts, but keeps the DNA. That these two fragments will be kept, and these two fragments, so the whole long genomic DNA will be kind of intact. It is as one molecule that has had TN5 fragmentation on it. So let's see here. Okay, so now let me see what we have on here is, okay, so we had TN5 genomic DNA. TN5 cuts the genomic DNA or fragments it, and then we attach the long genomic DNA to these barcoded beads. And as you see here, they call it tail bead or barcoded tail bead. So each of these millions of beads, they have a unique barcode, and as you see, it's the unique barcode, or the barcode is zoomed in here. And each of these beads will contain a specific or a unique barcode. Of course, not one molecule, it's probably millions of these barcodes are on the surface of this bead. In the article, they don't at all mention how they have generated these barcoded beads or tail beads, barcoded tail beads. So it's, to me at least, from the article, I cannot say or I cannot read anywhere in the article how they have generated these barcodes. I'm quite sure, or yeah, at least I don't believe that they have synthesized 10 million different 10 million different probes and attach them to different beads. I could speculate how they have done it, but I will not speculate on in here because yeah. Well, I don't want to speculate, but in our Zoom sessions, for example, I could maybe discuss this with you. And why I'm not speculating is because it's not fact. I don't know if it is really in that way they are generating these barcodes on the beads. Okay, so just accept that they have these barcoded tail beads. The next step in this, or the next thing that happens in this first step is that they attach the one end of the adapters that we have in these TN5 molecules. They will attach them to these barcodes, and even here they haven't exactly revealed how they are doing it. The only thing that in the supplementary you will see that it's they have used three methods, but this method one is the one that has been used to generate the data in this article. And they say that they are using a ligase to ligate the end of this to end of this, or 5' end of this to 3' end of the barcodes. And they haven't revealed anything about which sequences they are using for ligation and so on, but even here we accept that they are doing a ligation here to attach the TN5 molecules to the surface of these beads. And obviously here we have a long DNA molecule that goes probably around this beat, uh, around 10 to 100 kb, and here they are showing only a part of this long genomic DNA. And as I mentioned, this is around 600 bases. Okay, so after coupling the TN5 to the surface of beads, they add another TN5, but this time this TN5 has a different adapter. This one on the bead now has the blue adapter, and this one has the red adapter, and this will cut in between here. So if it cuts just in the middle of this fragment, we are generating 300 bases on this side and 300 bases on this side. And obviously it's just random, so it could be 100 bases on this side and 500 bases on this side. And sometimes you maybe don't get tegmentation, only one tegmentation here. It is possible that you would get two tegmentations. But from this picture, they are showing only one tegmentation in the middle here, so around 300 bases on each side of this. All right, so now we have DNA molecules on the surface of the bead, and each one has one adapter here, and these are Illumina's sequencing adapters. So the next step is they add, for example, SDS to denature the enzyme that we had, or the enzymes that we have here. So the enzyme will be removed, and what will be left is one end with this adapter, and we have this adapter on here. We have the barcode on one end, and then we can do PCR on these. So in the PCR, we are using Illumina's primers, P5 and P7. They're calling it these primers P7 and P5. I think this picture is not completely correct. So I think here at the end here, at the 5' end, we have this green sequence for to be used as a universal primer for amplification. On this side we have this red one, so P5 could be, for example, we are using this, and then here is this part is P5. All right, so we amplify and get a library that has this construct. We have P5 and P7 at the ends, genomic insert around 100 to 300 bases. Then we have sequencing adapters here, and then we have two indexes. If you remember, in Illumina you could use two indexes. For some applications one index is more than enough, but in this case they are using two indexes. But this index 1, is actually our barcode. So this is not really an index. It's 18 bases of our barcode here. So what we do is first we read we sequence in this direction, read 1, then we read, and this is around 140 bases. and then 18 bases for the index. Then we do sequence read 2, and after that we sequence index number 2 and this will be our sample index, while this one is our barcode. Okay, and after this we do sequencing and data analysis. And this is something like, and it's something like this we get from the after we have done the data analysis. We have reads that we can map to a reference genome. In this case, is human genome. And as you see, these have been mapped to a specific region. And why they have been in one line here is because all the reads in here have exactly the same barcode. So in this way, by reading the barcode, we can track back that they are from one long genomic DNA. Okay, and this is another barcode, and these are the reads that we have got from that long genomic DNA. Maybe 10, 15, 20 reads per long genomic DNA. And we are using this information to resolve the haplotype. And let me make this picture a little bit bigger and try to explain how we resolve the haplotype by reconstructing long reads and then mapping them to the reference genome. The haplotyping actually is done by making use of SNVs, or single nucleotide variants. So let's say that on this fragment we have here we have an A. Here on this position we have C. Let me see. This one we have an A even here, and then on this we have a G. All right. These are SNPs or SNVs that may exist in this individual. Okay, so the reads from this barcode have also, some of them have been mapped into the same region as this one. Well, okay. Let's say that even here we have a T. Okay, so in this we are reading on this sequence. We are reading even here a T. But here we don't have any read or any. We haven't covered this sequence where we have a C here. And let's say that we have on this one we have a T even here, but here we have covered this part, so we are reading T on this sequence. This one has not been covered, and this one has not been covered either. And then we go to this sequence. Here we have covered this part, so we are reading even here a T. This has been covered, so a C here. Oh, nice. This one is a T. Here we have an A. But this fragment was shorter than this one, so we are not covering these two reads, and obviously we cannot read this G on this barcode. Okay, so let's go from here to this one, and now we have T here, we see a C here, and nothing else here. Okay, and on this, on the other hand, we are reading in this position and this position. This is an SNV that could be A to C, and it is in this part or this C, in this individual we have a heterozygote. And let's say that this is an A to C, this position, and then this one is T to C again. This one is C to T. This is T to, let's imagine it's A. And then we continue here. We have A to G here. And even this one has been covered here, G to T. Okay, so all other reads, or all other sequences or bases in these reads are similar to this one, but this C here, and even C here is here, up here we have T, and here it's a C. We continue with this barcode, and here we see a C. We haven't covered this one, we haven't covered this one, but this one is covered here, so it's a G, and that's it. And for this, we see a C here, a C here. This is not covered, this is not covered. This we have a G, and here let's say that these two are the same. No, okay, we don't have anything here. Okay, so based on these SNVs, we can see that the reads from this barcode and the reads from this barcode and from this one and even this one are coming from one haplotype. And we can connect all other reads or from other barcodes in the same way. So we can continue on this direction the whole way. So what we can see is that these belong to one haplotype. These three are all reads from different barcodes with in down here are from one haplotype. So that's how you do the haplotyping, and resolving the haplotypes is called resolving the haplotypes. And as I said, the same way we continue the whole way and do this for a whole chromosome. And sometimes we will get gaps, and then the phase block may become around. In this case, they have here in this table we have some stats, and we have generated phase blocks, N50 phase blocks that are around 60 million, 60 million bases. When they have this. They have sequenced this many clusters. When you go down in sequencing depth, N50 phase block drops a little bit, or yeah, is half of this, and this is around half of this. But this N50 phase block of 60 mega bases, it's quite decent. It's very good. Okay, are there any problems? Because this seems to be a very nice technology, very easy to perform. The whole process here is around 3-4 hours. The only thing that I can think of is, apart from the problem that I maybe mentioned here, that when we cut here, we are never cutting exactly in the middle, so we are not generating around 300 bases and 300 bases. Usually we have observed at NGI, they have National Genomic Infrastructure at SciLife Lab. They have tested this method, and the insert size are quite short, 100 bases, 120 bases. So apart from this, the only big problem is that in read 1, we are here the sequencing 140 bases, and then we save 10 bases for reading this, because this index is 18 bases and Illumina's indexes are 8 bases. So we are, instead of 150 cycles or reading 150 bases, here we do 140 and save 10 cycles for sequencing these 18 bases as index. 8 for, usually we use 8 bases for reading the Illumina index, and then 10 other for, so in total 18 bases. And then in read 2, it's 150 cycles, or reading 150 bases. What that means is that we usually, different users of Illumina's sequencing, they would like to use or read 150 bases. So if you have TEL-seq libraries for sequencing, you have to use the whole flow cell of for Illumina's flow cell, and the flow cell on, for example, NovaSeq has 4 lanes, and each lane, the cost for sequencing only the kit is in 42,000 Swedish kronor. And so each lane 42,000, so times 4, it's around 170,000 kronor for sequencing. And you cannot combine TEL-seq libraries with any other type of libraries, because here you are reading 140 plus 18. Common libraries, they are reading 150 plus 8. So you have to use the whole flow cell for TEL-seq libraries, which means that you must prepare a lot of libraries from different samples in order to use a whole flow cell, and on each lane of this flow cell, you usually sequence 4 samples. So you need at least 14 to 16 samples for sequencing. So this is a quite a big problem. You cannot combine TEL-seq libraries with other libraries for sequencing. Okay, so and here they are showing how they have done the haplotyping, haplotype 1, haplotype 2, and as you see here we have a deletion. But it's interesting that even haplotype 1 has a partial deletion. So in this individual we have a partial homozygous deletion, and then here we have heterozygous deletion. All right, that was the last slide. This is the same question as on project 12. So compare and contrast the two haplotyping methods in these two projects, project 6 and 12, and also discuss the use of long-read sequencing platforms for doing haplotyping. Okay, so this is our discussion topic when we meet.